Hi everyone, thanks for joining me. What I'd like to do with this short video is give you a brief introduction to the Oticoli project, which was the 2006 MIT iGEM team project that they presented at the iGEM Jamboree. This little video lesson is not intended to replace, a chance to watch them give their presentation at the Jamboree. That video is available and really worth watching. That team had some real rock stars on it, and it's fun to watch them present. Another complimentary video lesson for this one is the biodesign short lesson that will walk through some other examples of the abstraction hierarchy, moving from systems to devices to parts and then back up again through that hierarchy. But this lesson should give just a brief introduction of how the Oticoli project was designed and some of the details of that design, and hopefully will be a nice introduction to the That Smell Lab should you be doing that one that's on the BioBuilder site. So I'll begin by just mentioning that the iGEM team had a very broad system level description initially. They thought of the cell as a black box which could receive some inputs and generate some outputs, and they dreamed up what they might like as an input and what they might like as an output. And the system that they came up with was this one, that a precursor compound might be added to the media that the cells were growing in, and out would come from that black box of a cell some smell that would be more pleasant than the traditional bacterial smell that people in the lab can recognize a mile away. So with this as their initial system level description, they realized that they would need some devices within that bacterial cell to generate those smells. The devices that they chose, after eliminating a couple of others, were a wintergreen generating device, which made a minty smell, and a banana smell generating device, which made this media smell like bananas. And with these devices in place and their ideas starting to formalize, they realized that one way to really regulate this smell production that might be very interesting is that rather than have the smells produced when you finally add the precursor compound, have actually the different stages of cell growth regulate the smells that were produced. So if you think of a traditional bacterial growth curve, which is what's shown on the bottom left of this slide, a very dense culture of bacteria, if you take a small amount of that and add it to some fresh media, it will go through what's called a lag phase of growth. The cells are sort of waking up from their very dense growth. They hadn't been dead, but they also hadn't been actively growing. So during that lag phase of growth, there is a lot going on in the cell, but there's no increase in cell mass. So you couldn't really measure an increase in optical density if you measured it in the spectrophotometer. So the lag phase, as you see on that bottom left graph, is a flat is a time. Time is shown on the x-axis. Over time, there is no increase in cell mass. But once the cells sort of wake up and get going, they reach what's called the exponential phase of growth, or the log phase of growth. And this is the very rapid phase of growth that bacteria can undertake when nutrients are plentiful and that they have been actively growing. They can double on the order of 30 minutes, a doubling every 30 minutes. But that doesn't last forever, and after another period of time, the nutrients in the media get used up, the cells get crowded, they start secreting waste products into the media that inhibits their growth. And so they reach that third stage of growth called the stationary phase of growth. And that's where a plateau is reached in terms of the cell mass. You see over time, the number of cells does not increase, cell mass does not increase, optical density does not increase. But the cells aren't dead, they're just sort of in a stasis, in a suspended state. And again, if you took a small number of them and re-inoculated them or subcultured them into some fresh media, they would undergo this lag, log, and stationary phase cycle all over again. So now looking at the graph that's on the bottom right of the slide here, the idea of this iGEM team was to regulate the production of these smells with the different stages of the cell growth cycle. So what they chose to do was have the banana smell generated during the stationary phase of growth, and they wanted the wintergreen smell to be generated during the log phase of growth and to disappear during the stationary phase. So to do that, what they needed to do was bring in the precursor compound generating devices into the cell. So you remember in their initial description, they had the precursor compounds simply being added to the cell by some experimenter. Here now, what they're asking the cell to do is generate these precursor compounds on their own. So there is now on the device list a salicylic acid generating device. Salicylic acid is the precursor for the wintergreen smell generating device. And then there is also an isoamyl alcohol generating device because isoamyl alcohol is the precursor to the banana smell generating device. And so with the cell able to make these precursor molecules, they can produce the smell based on their cell growth cycle if we now can regulate the wintergreen generating device and the banana generating device by cell growth. And so how the iGEM team chose to do this is described in the parts level specification for their system. We'll look at the banana smell generating device first, and what the team chose to do was to identify a promoter of transcription called the OSMY promoter, which is only active during the stationary phase of growth. And so by connecting the banana generating device to the OSMY promoter, this stationary phase promoter, they were able to produce the banana smell during the stationary phase of growth. Now, certainly they could have found a log phase promoter that might have turned the wintergreen smell on during the log phase and generated something similar to this structure in order to specify their log phase wintergreen smell. But instead, they chose a different route. They chose to take the OSMY promoter, which is the stationary phase promoter that they knew was working, and connect it to the wintergreen generating device. But between those two parts, they included something called a genetic inverter. And the logic for the genetic inverter is shown at the very bottom right of this slide. The idea is that when the stationary phase is on, the OSMY promoter is on, then a log phase promoter would be off. So if it's a log phase promoter driving the wintergreen generating device, it will turn it off. Whereas if you add the OSMY promoter and an inverter when stationary phase is off, the product will be off in stationary phase and it will be on in the log phase of growth. So the idea is that this inverter, this genetic inverter, will switch the logic during stationary phase. If you're in stationary phase, then the downstream device is off if it's log phase, and if stationary phase is off, then you're measuring something in log phase, that should be turned on. So I'm not going to tell you precisely how that inverter works. We can look at that when we start looking at a parts level description of different elements, but it was a reasonably clever design that they used. So the precursors were made by the cell. The OSMY promoter, which is active during stationary phase, is either turning the banana smell generating device on during stationary phase or is turning on an inverter and therefore turning off the wintergreen smell during stationary phase, making it on during log phase. And then the very last modification that the iGEM team made to their system was to modify the E. coli chassis, the system in which this genetic program is running. And the modification that they made was to remove indole production from the genome of that cell, because it is that indole smell that is the stinky smell that you often associate with bacteria. And so by making this a scentless chassis, you can better smell the wintergreen and the banana smells from this synthetic system. And so their final system is shown here. It has a modification to the chassis which removes indole. It has the precursor production devices, the salicylic acid and the isoamyl alcohol generating devices. And then it has stationary phase promoters driving either the banana smell generator or the wintergreen smell generator, but the wintergreen smell generator has an inverter device between the stationary phase promoter and the wintergreen generating device, making the logic inverted. So during stationary phase, wintergreen is off, but when stationary phase is off, wintergreen is on. So this is their very clever project. I hope it's a nice introduction to their Jamboree presentation if you have an opportunity to watch that. There is also, as I said, a lab on the BioBuilder site called Oh That Smell, which allows you to work with their system, at least the banana smell generating aspects of their system, and also try to regulate production of banana smell in log phase using inverters. I hope this has been helpful. I hope it's interesting. Good luck and have fun.